Humans have always been fascinated by the idea of alien life outside our galaxy. The mystery of the unknown world has both captivated and frightened us. But perhaps the biggest threat from the unknown comes in an alien life form found a little closer to home. Bacteria are single-celled organisms that have been among us since the beginning of time. Often, we coexist peacefully with this tiny organism, but recently, outbreaks of antibiotic-resistant strains have caused a mild panic in the healthcare industry. Scientists are racing against time to find new antibiotics to combat these persistent and almost alien organisms threatening human life. Bacteria are everywhere. There are over 300 kinds of bacteria that exist in the human mouth alone. They exist in our hair, on our skin, and in our homes. There are many mysteries surrounding these common, single-celled organisms, and research is being done to develop new toxins that can kill harmful bacteria. Antibiotics were an astonishing addition to modern medicine. In 1928, Alexander Fleming discovered penicillin, the first antibiotic. Since then, many new antibiotics have been invented and put into use in today's medical fields to combat harmful bacteria. However, these antibiotics are no longer working with the same potency as they did 20 years ago. Bacteria are getting stronger and more resistant to the antibiotics designed to defeat them. Antibiotic resistance is not a recent discovery. A study done in 1943 confirmed that bacteria are able to adapt and survive attacks from toxins. Two groundbreaking scientists, Luria and Delbruck, realized that when bacteria were introduced to a toxin, large numbers of the bacteria were killed off, but some bacteria survived. They decided to look at a series of 5 to 100 cultures of bacteria that were introduced to a toxin. After 24 and 48 hours, they would count the number of bacteria still present in the cultures. The study showed that bacteria in their cultures were surviving in randomly dispersed clonal groups, small clusters that had formed after all the other, weaker bacteria had been killed off by the toxin. Today, we know that bacteria reproduce by creating exact copies of themselves, or clones. The surviving bacteria left after 24 and 48 hours stemmed from mutant bacteria that had passed on their mutation in the clones created in bacterial replication. The study showed that bacteria are capable of mutations, meaning that they have DNA and are able to pass on their genetic differences, which make them resistant through reproduction. Mutants are bacteria that have a genetic difference from other, non-resistant bacteria. Bacteria with the ability to survive antibiotics can pass on their ability through bacterial replication. This is a process in which a single bacterial cell creates two identical copies of itself. These two cells will have exactly the same structures as the original cell, including its DNA. DNA is a structure within the bacterial cell that contains unique genetic code. Bacteria have the ability to pass on the traits that allow them to survive because they have DNA with unique genetic codes, some of which contain mutations that make bacteria strong enough to resist toxins, including antibiotics. Bacteria replicate through a process known as binary fission. In binary fission, a single organism, in this case a single bacterial cell, creates enough DNA to be shared between two cells. The first step in binary fission is a process known as DNA replication. In DNA replication, the bacteria cell is going to make copies of its DNA and to be passed on to two new identical cells. DNA replication takes place in the nucleoid, the structure of the cell that contains DNA. Inside the nucleoid, the cell makes many identical copies of the cell's DNA. Once the strands of DNA have developed, they migrate to opposite sides of the nucleoid. Once both sides of the cell have the same amounts of DNA, the cell splits, creating two new cells called daughter cells. Each of the daughter cells contain exactly the same amount of DNA that the original cell started with, as well as all of the same traits contained in the original bacterial cell. In this way, bacteria are able to pass on their mutations. Because the daughter cells are identical copies, they have exactly the same kinds of mutations as the original cells from which they divided. Bacteria are able to go through this process of replication multiple times, and they do so very quickly. Because the mutants can replicate so quickly, resistance to antibiotics can be passed on to new bacteria in a matter of years, making new antibiotics ineffective and outdated. As modern scientists continue to struggle with developing new antibiotics and methods for combating these new resistant bacteria, exploration into other mysteries continue. Who knows, maybe our space explorations will lead to a new discovery that will solve all our problems with bacteria here on Earth. Only further investigation into the world's many mysteries will reveal the truth.